This infographic shows a popular biological concept. Long telomere caps protect young DNA, and as cells divide over a lifetime, those caps wear down until the cell becomes old. This simple visual correlation has driven a massive assumption in commercial biotech. The logic goes that if short telomeres cause cellular aging, then therapeutic telomere lengthening must be the definitive path to reversing aging. But viewing telomeres solely as a biological clock ignores why they evolved. Continuous, unchecked cellular replication is the exact definition of cancer. We tend to look at the physical decline of aging and assume the mechanism driving it is a design flaw. We see the wearing down of our cellular machinery as an error to be corrected. The data tells a very different story. The gradual shortening of your telomeres is not a biological mistake. It is a highly conserved, violently efficient tumor suppression mechanism. Pursuing therapies to permanently lengthen telomeres without respecting this evolutionary purpose risks removing the very safeguard keeping our cells from mutating into deadly tumors. To see this in action, we have to look across the mammalian class. Researchers recently analyzed phylogenetic data spanning 57 distinct species of mammals to map out how telomere dynamics affect survival. This chart plots a species' baseline telomere length on the horizontal axis against its historical cancer incidence on the vertical axis. If long telomeres were purely protective, we'd expect a downward trend. Instead, the data forms a distinct, undeniable upward line. Across mammals, longer baseline telomeres reliably predict a higher risk of developing neoplasia or cancer. This completely inverts the framework marketed by commercial anti-aging clinics. In the wild, maximum telomere length does not equate to maximum health. Evolution actively selects against long telomeres in many survival contexts. To understand why, we have to look at what specific problem a shortened telomere solves. Consider the massive physical differences between a mouse and a whale. A whale has trillions more cells, and those cells divide over a lifespan measured in decades, not months. Every single cell division carries a baseline risk of genetic mutation. An organism with the cellular volume and lifespan of a whale should statistically be guaranteed to develop cancer early in its life. Yet they don't. Large mammals like whales do not experience higher cancer rates than mice. This phenomenon is known as Peto's paradox. The solution lies in their genetics. Large mammals evolved uniquely shorter baseline telomeres compared to smaller species. Alongside these shortened paths, large mammals also strictly repressed the expression of telomerase, the enzyme that rebuilds telomerase, in their somatic cells, enforcing firm limits on how many times a cell can divide. Short telomerase act as an evolutionary tax. They impose a strict limit on cellular replication that massive organisms must pay to avoid cancer long enough to reproduce. This cap on replication is called the Hayflick limit. For normal human cells, it restricts division to between 40 and 60 cycles, physically enforced by the gradual erosion of the telomere caps. This diagram illustrates exactly how that checkpoint functions. Once the telomere ends wear down to a critical threshold, tumor suppressor proteins like p53 read the short telomere as DNA damage and force the cell into senescence, permanently halting its division. Acting as the guardian of the genome, p53 ensures that cells with critically short, unprotected telomeres enter a dormant retirement, rather than continuing to replicate damaged DNA. The danger arises when a cell acquires a mutation that disables these specific tumor suppression pathways. Without p53 or the retinoblastoma protein functioning correctly, the cell ignores the senescence checkpoint. This pushes the cell into a state called telomere crisis. The cell goes rogue, continuing to divide even though it has entirely depleted its protective chromosomal caps. Bypassing the senescence checkpoint converts a stable, aging cell into a driver of systemic genomic collapse. This illustration shows the mechanical consequence of telomere crisis, the breakage-fusion bridge cycle. It begins when two chromosomes entirely lose their caps. Without telomeres, the cell mistakenly fuses these sticky, exposed chromosomes into one massive structure. The true crisis occurs during anaphase, when the cell divides. The fused chromosomes form a physical bridge, stretched to their structural limit. This microscopic image captures that exact physical tension occurring inside a real dividing cell. The fluorescent green chromosomal mass has formed a literal bridge, caught and stretched between the separating red spindle fibers. Eventually, the forces of cell division violently tear this bridge apart, shattering the DNA and randomly distributing broken fragments into the new daughter cells. Short telomeres, without a functioning senescence checkpoint, actively drive the massive chromosomal rearrangements and mutations that fuel early oncogenesis. The genetic chaos created by the breakage-fusion bridge cycle is highly destructive. If the cell continues to blindly fuse and shred its chromosomes with every division, the accumulating damage will eventually cause the cell to self-destruct. To survive, a developing tumor must stabilize its genome. This progression is the biphasic model of cancer. After acquiring necessary mutations during the crisis phase, the tumor deploys a decisive stabilization mechanism. It upregulates the TERT gene, successfully reactivating the telomerase enzyme. Clinical data shows that 85 to 90% of all cancers manage to reactivate telomerase. This allows them to rebuild their chromosomal caps, immediately halting the shredding cycle. Telomerase does not initiate the cancer. Instead, it rescues the highly mutated survivor of the crisis phase, locking in its malignant traits and granting it unlimited replicative immortality. This represents a core contradiction in tumor biology. Cancer needs the instability of short telomeres to rapidly mutate, but requires the protection of telomerase to survive long-term. We can see just how sensitive this balance is by looking at what happens when natural selective pressures are removed entirely. Returning to the phylogenetic data, researchers found a striking pattern regarding artificial selection. Domesticated strains of mammals consistently possess significantly longer telomeres than their exact wild counterparts. When human intervention removes constant threats like starvation or predation, the organism is highly likely to live to an old age. In that protected environment, the evolutionary need to strictly suppress tumors via short telomeres begins to relax. This domestication effect proves that telomere length is not a static biological clock. It is a highly reactive evolutionary variable, constantly adjusting the balance between healthy tissue renewal and lethal cancer risk. Therapeutic interventions that universally aim to lengthen human telomeres misunderstand this deeply conserved biological system. Longevity science requires maintaining the precarious, ancient evolutionary balance that keeps our cells from achieving malignant immortality.